Good morning, everyone. I would like to thank the organizers for having me and also benefiting from my previous speaker. So we will talk about more or less the same things from another angle. Inflammation and inflammatory cytokines turned out to be relevant to aging. But how does this work? And is it really reversible? You will see the results are quite confusing. So I'm busy since a long time as a hematologist and a hemorrhagiologist with many prestigious institutions in Europe and Asia, trying to understand this and also trying to come up with something that would be anti-inflammatory in the long run and not toxic. This was always my real fundamental preoccupation. So TNF, we have heard everything about, is in fact inducing, among a bunch of other actions, adhesive molecule expression on the famous endothelial cells Lou Hawthorne ended up with. And here you see them, so I have to do this here and not here, yes. Here you see this little red foot here, who stands up and recruits activated leukocytes by TNF, by the way, in order to drag them into the tissue and perform their immune function. That's how it works. Yet, this process can be completely submitted to sabotage. By whom? By cytoskeleton. Cytoskeleton of endothelial cells is deciding if the foot goes up or not. And if you have this picture induced by TNF-alpha, the foot goes up and can recruit the adhesive molecule, and can recruit the activated leukocytes. But the process is submitted to regulation, and eventually you cannot drag up the foot. So this is inflammation. If you don't drag up the foot and don't recruit the activated leukocyte, you don't have inflammation. So this was quite clear for us at some point very early in the story. And we tried to understand what's going on with this actin fiber polymerization in the endothelial cells. And we discovered that they are mediated by a family you all know about, RTPases. And this cell adhesion molecules expression is set in motion concomitantly. So that raw GTPases controls the cell shape, obviously this was known, and motility, but also regulates actin cytoskeleton for this famous signaling, mechanotransduction of the signal. You have to understand that endothelial cells are in a very particular environment. No other cell in the body is. That is blood flow. Hence, my hemorrhagical background was here really gratifying me and understanding a lot of peculiarities of this signal transduction. So we started at some point with my team at the Necker Hospital in Paris, where I met Aubrey in '96, suddenly in the floor, coming with this enthusiasm towards us. We were looking at this guy saying, where is he coming out from? And started together with the CNRS and the other institutes specialized in extracts of natural plants in Asia, notably with the colleagues of the University of Hanoi, that were tracking with ethnobotanists extracts of plants in the rainforest, healing plants, just healing plants, no further specification. And so this rare plant called Alford-Jackendack, according to a British general passing there, not even classified in botanic taxonomy, turned out to contain a molecule that did the job, and I will specify what was the job. But in fact, it was plenty of precursors and metabolites, so that quantum physics fellow once told me, you don't have identified a molecule, you have identified a frequency, which is very interesting because I think that quantum physics is a forgotten aspect of biology. So to make the long story short, this monoterpene, it turned out to be a monoterpene, but anyway, molecules have a class, and discovered in the rainforest. And finally, as you can't really drain plants from the rainforest in Vietnam, when we had to test it in first in human studies, we found back the same molecular complex in the skin of the orange. This is called Genus botanicus. And Genus botanicus is a scientific compilation codifying precisely all medicinal properties of plants. I didn't obviously know anything about all this. So, as other drugs, D-limonene is a farnesyl transferase inhibitor. And in fact, at the same time I was tracking the possible mechanism of action of this molecule, another lady, a chemist at the Wisconsin University in the US, at a higher dosage, still not toxic, was characterizing it as a very important tool to stop the progression of pancreas, gut, and breast cancer. So I thought, very strange, because at those concentrations, we had a strong pro-inflammatory effect. But we were looking at an anti-inflammatory effect. So, to wrap it all together for this first phase, ROA1 is, in fact, installing a completely different way of a molecule to function that we could define biophysical regulator, negative regulator, of pro-inflammatory response. Here in this cartoon that you have already seen, I have added the circulating tumor cells. So you will ask, but how does the tumor cell get into the tissue? Is it not recognized? No, it's not recognized by the immune system because it's wearing a cap, mimicking to be a leukocyte, and particularly mimicking to be an activated leukocyte. So normally it is dragged into the tissue. And in fact, this makes understandable the results of Pamela Crowell, who was not able to stop cancer, but who was able to reduce the mass of the cancer lesion and to stop cancer dissemination, which is metastasis. It's exactly this mechanism. So, to resume this first part, when we can say that inflammation mimicked by TNF transduces into the activation of leukocytes, which need the expression of adhesive molecule to be dragged into the tissue, we also are conscious that this diapedesis to succeed need the polymerization of actin fibers. And these actin fibers in the endothelium can be stopped by a molecule that is just operating the inhibition of ROA1. And this is just the mechanism. Is it reproducible in other cell types or in other conditions? We'll see. For the first time, we did a co-distribution and characterized that the phenomenon of actin polymerization was co-distributed with the phenomenon of superexpression of adhesion molecule and that it was reversible. But how does this stand now to the fact that in human studies, we were able to see an impressive inhibition of interleukin-6 especially after what we heard from Lou about interleukin-6. So which one had played the inhibition of interleukin-6, which is a very interesting molecule because it's also a myokine. It's not only a cytokine and has several positive and negative, good and bad roles. It's completely ambivalent. So we published all this work and also submitted it to the SASP model of Judith Campisi, only to discover that a reversibility, so it was not at all a senolytic, it was really a xenomorphic compound. Yet, the inhibition rate was intriguing, because at that time, AstraZeneca was playing with monoclonal antibodies to inhibit ICAM1 or VCAM1 at 20%. And we had immediately these huge inhibitions. So we dragged further and looked at, obviously, NF-kappa-B. Not personally, not to be influenced in our results, so we collaborated with the IGBE in Naples, who were specialized in NF-kappa-B, and got and published the result. So taking all this together, when we started a few years later, the first in-human studies, together with Claudio Franceschi, in a European project, we had already a lot of insight. Others will come later, but about inflammaging and inflammation simply. What was interesting about this study is that the 65, 85-year-old probendi were not sick. They had no heart issues, they had no cancer, no hypertension, no depression, no obesity, but they were all pretty well inflamed. And we were able to show that, in fact, here the inflammation for the first time was expanding a little bit into the anti-inflammation of our lead, into the fibrinogen. Fibrinogen speaks about the thrombosis. I mean, we all die from thrombosis. If I can resume what Martin has said in the beginning, why don't we study cardiac infarction? Because we all die from thrombosis. So the problem is fibrinogen or sepsis, as Lou put it, but also HOMA-IR and insulin. So we are into cardiometabolic disease here. And this was quite interesting to us. But where is chronic inflammation coming from? Big question. 20 years ago, 10 years ago, nobody had an answer. Today it's quite clear. Social stress contributes to a multi-scale inflammatory map, leading to polycrisis and organ dysfunction. And in fact, you have a phenomenon that is called cortisol bathing. We are all into cortisol bathing, due to cortisol hypersecretion and resulting in a leaky gut that is responsible for the dysbiosis. And dysbiosis is causing a lot of damage. Now, main drivers, accelerating aging, can only work if the gut barrier is leaking. That is a monumental universal truth. But it's very difficult to track. And in fact, can nutraceutical supplements or prebiotic fibers counteract this because of epigenetic modification inducing anti-inflammatory and tumor suppressor gene expression? We simply showed that this monoterpene were able to restore the gut barrier and to reverse the ratio between good and bad bacterial strains, which is important in the gut-brain communication. When it comes to other properties of this small molecule that is considered by a whole bunch of people as a simple dopaminergic molecule because it enhances dopamine in the brain, this is even more interesting because in middle age, dopamine slows down. And why is that? Because neurons quench dopamine, their own dopamine, with the same mechanism as endothelial cells drag up the arm of the adhesive molecules. They need an inside elasticity at the side of the synapses in order to do this. And when they are just stretched by inflammation, our own dopamine is stuck into the synapses. So in fact, what we see here is just the effect of the monoterpene, that it is stopping the stress fiber formation. We had, concomitantly to these Japanese researchers who published 2 months before us, that's live, undergone very interesting studies together with some ethologists showing the change in behavior in rats and showing their high motivational effects and lower irritability. So that we concluded that we had quite a point here. Mood amelioration via increased dopamine is just part of the whole picture and was also characterized in humans with a little gender issue. This is typical for natural substances. They are either like xanthines, favorable to men or to women, such as terpenes. We wrapped up all this. The whole story of the varicosities towards the neurons is very interesting, but I see I have not so much time anymore. So we go into the lifestyle story. And Eric Verdin was saying once that lifestyle is 93% of the prevention of aging. So what are the characteristics of healthy lifestyle? Can we be reassured by visible effects? Do we see it? What about lifestyle when it comes to geroprotection? We all know about this great enterprise by Wellcome about resilience. But resilience is based on what? Physically, from the physiological point of view. We have some ideas about that, and we think that vagal activation is a longevity-promoting tool, and that it can found resilience. You can't be resilient if you are super excited. So, cardiac coherence, rhythmic breathing, inhaling, and also trying to learn to meditate much contribute to the anti-inflammatory endogenous activation of the vagal system. And so, once you get the pretty picture of those ruling the world, they're currently having a hard time. There's some guy out there doing NF-kappa-B inhibition, doing TNF-alpha interleukin-6, interleukin-1 inhibition, doing gut barrier repair, doing microbiota repair, ratio reshape, and mood modulation thanks to vagal activation. That's a mess. We have to take control again. And so introducing the first in human results on skin, in fact, fisetin and quercetin have been characterized as anti-inflammatory, but senolytics first of all, which many supplements claim in a polypharmacologic way, and by the way, from the pharmacogenomics is a nonsense because 95% of us have a negative affect treatment due to the phenotype. So Merete Hardestal was interested into the impact of anti-aging therapies in cancer promotion. This is a very classical subject, and she was able to show, which is very interesting for us, how compared to the classical cytotoxic chemotherapy, quercetin and fisetin were, of course, extremely efficient in senolytic activity, but also anticipating by 2 years the appearance of skin cancer. So I think we have to be very conscious about this play that also has been addressed by Lou about the concomitance of cancer and anti-senescence drugs. We did go into resetting of visible skin lesions like this one in psoriasis, and I'm happy to announce that the famous quality of life aspects were also visible. In particular, when it came to itching, here we have a testimonial of a Formula One champ who even says his skin was clearer. I think this is just touching, a man who tells you his skin is clearer. And this lady with her terrible itching, all this was always very quickly. And here the famous elasticity effect that I affection very much, because no aesthetic medicine intervention can ever produce this. So you need something more than only a molecule doing something. You need a whole complex contributing to the effect. This here is only to say that theoretically, every TNF-alpha inhibitor should be able to restore wrinkles. Social stress can also hit young skin, as we see here in atopic dermatitis, and we also checked about the leaky gut. And after 10 days, this was perfectly restored. In the sense also of a rejuvenation, I would say. The last one concerns aesthetic medicine that is producing many side effects. And again, only repairing the gut barrier, it's always the gut barrier. In 10 days, you have this shift from a completely devastated mouth region, very delicate region, to an original, attractive region to just be able to pass on television for this actress. I will not name her. But Delimonen, is it an upcoming small molecule of the future? Anti-inflammatory, xenomorphic, mood-enhancing, proven to be safe, systemic improvement of the gut barrier connection. I like this very much, but also the skin-brain connection. A miracle remedy? No, just a science-backed up product line for prevention and remediation, a most precious sustainable companion for healthy longevity. So at some point, Alexei Moskalev, who is in this conference, was talking about terpenoids and geroprotection and quoted all my papers. And I said, oh my God, I am a geroprotector now. It's fantastic. I have a name. I have a category. I am there. I'm in. And so I even discovered that the Academy of Sciences of Argentina even published a paper on monoterpenes and cytokines. And so as it was the COVID and there was a lot of time, I decided to tell my own story in this review. But today, there is an incredible increase, here you see it, of Delimonen science, a fourfold publication increase. And here you see the conundrum. And I think that as this is just the review of the publications, that in a few years, this picture will drain massively neuroprotection here into the anti-inflammatory effect. And some ideas were clearly given by Lou precedently. So, as I have 37 seconds, I just can acknowledge my really affectionate group who made it possible to address with the art of the moment the question, why does inflammaging occur in the Ristomed study? And by the sequence, I also would like to address the fidelity to China that was not dependent on me, but it's now 20 years we interact. First with the Fudan University of Shanghai, who stayed in my lab for nearly 8 years, and then with now the 2nd military hospital in Beijing, who named me their scientific supervisor for their stem cell department therapeutics. And also sometimes I look into Singapore and a little bit farther away from central China. Look up my Instagram page. That was a revolution to me, and I'm very proud of it, where I try to break down all this. Time's up. Okay, thank you for your attention.
Can we just take a couple of questions, please?
Wow, 25. Perfect. No questions. Everything's clear.
Patrizia, Patrizia, that was wonderful. Obviously, I'm very aligned with everything you're saying about TNF and inflammation. There's one point you made, which I think was particularly courageous, which is talking about the role of stress as a trigger for all of these dysfunctional cascades. I'm wondering, though, what I personally think stress should be managed and measured as carefully as cholesterol. I'm wondering what tests could be done to determine when someone has crossed the line at a molecular level of stress and needs to do an intervention, whether it's yours or a lifestyle intervention.
Yeah, very nice question. The point is that stress is not obedient to our scientific rules, you see. It's just to be approached in a completely different way. To understand stress, you have to include the role of quantum physics, you have to include very measurable effects of the environment, you have to include the role of water in the body, you have to become much more interdisciplinary to understand how to deal with stress. It's really the top question of the moment. Very good question. Coming from you. So,
we just take one more.
Hello.
Hi.
To what degree can simple citrus peel substitute purchased pharmaceuticals?
Purchased pharmaceuticals. What is purchased pharmaceuticals?
Like, in other words, why would I go buy something in a bottle when I can just grate some orange peel into my salad?
Ah, okay. Oh, wait. It's just the therapeutic window. I mean, if you eat orange peel, which, by the way, is disgusting, how could you get the, I mean, not the whole wagon of orange peels. How could you meet the therapeutic window? We spent 3 years to define the therapeutic window.